Now that the other tools, which is helps to do quality check, the other tool is called MULTIQC, where it is an aggregative result to perform do reporting thing. We can get a fast QC report or fast speed report, Katada, PWA, star, all those things results we can able to visualize through MULTIQC, which is lately developed after 2020s. More people which is help using this to do a reporting purpose only. For main analysis, everyone using this fast QC only. So to perform MULTIQC, we need to install in the Linux system, and then we can able to perform the analysis. Similarly, we can able to perform MULTIQC in the Galaxy also. Next tool is called FastP. Okay, so far you can able to understand about FastQC, right? I hope you guys have understood about doing a quality check. Whatever I told in the graph, you can go into a Galaxy and you can able to see your report. So here our report is there, no? You can able to see our report over here. You can get all the graphs. So what I told, PUD base sequence quality, all the bar plot which is present in the box plot which is present in the green region, which is considered as a good, we can able to consider for the analysis, right? That is what mentioned over here. The quality is good and we can take the sample to perform the analysis. But in some cases, you can able to see the PUD based GC content is failed, a PUD sequence GC content is failed. It means the there may be more contaminations or the sample is below the condition. All will be happened over here. So we need to check all those. When we compare what are I tell in a theory, you can able to compare the results with of what we are doing it, and then we can understand of what that fast QC data report tells you to do analysis is. I hope you guys have understood. Okay. Then I'll go into the next tool. Next tool is called FastP. FastP is a tool to perform the pre-processing of a FastQ file. One second. A pre-processing of a FastQ file. What this FastP tool do is, we can able to do adapter trimming, we can able to do quality filtering, we can able to do length filtering, poly G trimming, duplicate removal, and it will provide the HTML generation report also. Why this FastP tool is using? It is a fast process which is multi-threaded. It is automatically we can able to detect the adapters which is built in QC reporting process. So the input for this is either we can give a paired end or a single end of the FastQ file data. So to run a FastP, we can run in the Galaxy, we can run in the command line. The command to perform is FastP. I is input read 1, small I is input read 1, capital I is input read 2. Output will get small OS that is trimmed, removed, quality filtered, cleaned data, and the next is read to the log file information. So these are the output will be provide, which is provide the clean FastQ file, which is where the remove the overrepresented data, remove the GC content, remove the adapter content, all will be there. And this data, the clean data will be taken as an input to perform the downstream analysis. That's why you are going to see in the coming up session, which is going to happen on July 13 and 14. You are going to see the next step of process, how to perform those analysis. So what is adapter trimming and quality filtering? So during sequencing, the adapters is appears at the end of the fragments, right? As I told in the beginning. So the adapters, if it is present while the sequence is there, it may be reduces the poor quality alignment. The alignment will be reduced. They help their the chance of getting a false positive variance also, that will reduce this the assembly quality also. So it will reduce the low quality filtering. The samples, the nucleotide reads, which is less than the Fred score value, less than 20, we'll need to cut that. That is where the adapter and filtering process is important. Another tool to perform the filtering, the other tool is called cut adapt as a command line based tool. We can use it in Galaxy also, where it is helpful to do microbial genome data, RNA data, or amplicon sequencing data, which is remove the library preparations, adapters, ligated, or low quality reads, all we can able to perform with that. So as I told, we need to remove the adapter contamination data. So where it do adapter trimming, primer trimming, quality trimming, length trimming, all will be possible. So we'll have the FASTQ data. We'll use the do filtering. We'll get the FASTQ, clean FASTQ file. And once the clean FASTQ file, we need to do a quality check again, whether the cleaning has been done properly, whether the trimming and quality filtration has been done properly or not. And then we need to do the next step of downstream analysis. That is the important steps to perform the NGS process. So to run the cut adapt, we can able to do the same step of process. So here I have mentioned the installation, and then I have mentioned the command line process steps to perform the same. Another tool, other important tool to do trimming and filtering, that is called a Trimomatic. This is also most widely using tool, which is very widely used from the researchers all over the world. The common function is, which is specifically for Illumina sequencing, that I need to mention, for to do pre-processing. It has have functions like adapter clipping, a slide window filtering, leading based filtering, trailing based filtering, minimum length filtering, all will be there. And the standard parameter of example to consider this all this, whether it is the sliding window which be 4:20 length, and the leading should be 20, trailing should be 20, less than the low quality, which is 50. All will be based on the specific algorithm to understand the average quality drop present at the particular sequence of what we are considering to perform the analysis. So each having the different algorithm, different thing to perform the understanding to do analysis of that. So what is read that is, during alignment process, the alignment is taken place based upon the locations of each position where the genomes are getting added. Sometimes if the adapter or the low quality reads presence, the alignment can't take in place properly. That leads to a false positive error. That leads to a low quality assembly process. We can able to identify the false variants. All this can be possible. For that, the read filtering, read length filtering is important to perform the downstream analysis. In the host contamination, that can be possible where whether sample or animal sample or sequence contamination, that is also possibly happen during the sequencing. It's all will be human error or machine error, everything will be there. We need to cut all those things and then we can able to perform those things. The other tool we can use do or call this is our latest advanced tool. So we can able to see the results before filtering and after filtering. Before filtering, the sequence looks like this. The quality is looks like this. After trimming and adapter contaminations, this is how the value looks like. This is far better. You can able to see, and you can able to take this data to perform the next step of analysis. So you can able to see the quality report in a better way to understand the matrices to before QC, after QC, how the read length, how the quality score, how the adapter content contaminations, what is the average quality. The quality before QC is Q27 only, but after QC it's Q35. That changes has been happened only due to a pre-processing step. And so this is how the report is of pre-processing done. We want to perform with any specific data. That's where the objective have given. We need to access and pre-process the Illumina patent E. coli data from NCBI SRA. The steps to this, you need to download the FASTQ file. You need to run FastQC, generate multiple QC if required, trimming using a FastQ, rerun the FastQC analysis and compare the results with the first raw data and the cleaned data. The output will be provided as a high quality data to perform the next step of analysis. Make it as your exercise to perform it for your analysis. And what are the problems we'll need to do, will cause, and what are the troubleshooting we need to consider. The problem is low quality will be there, high adapter content will be there, GC content biopsies will be happening, high duplication will be the chances, low coverages also will be there. All will be causes because of different condition. All will be having a solution to consider to change the data into a better way to perform our required analysis and get an output. So best practices methodology is we need to inspect the raw data to do a fast QC. Using fast QC, we need to summarize the report. We need to remove the raw quality reads, filter the reads, remove the host contamination, and after all this we need to do a fast QC check again, and we need to document all the parameters, reproduce it for the any set of data, and then which is consistent process to go with the another importance of analysis to get our exact biological output of what we have. So this is the hands-on exercise you can able to perform with that, and with that summarizing what I have teach so far is what how to download the data, what is FastQ file, what is FastQC, how to perform those, how to do filtering, all those things. So before summarizing this, I just want you guys to take into Galaxy again. So I told the tool to tools I have told you to do pre-processing. Can you guys, any of you guys tell what are the tools to do pre-processing? Okay, cut adapt is correct. Yeah. If you have, okay, I didn't see the few questions. Okay, you want me to do the quality of and I have influenza causes that is once, and we have been biases PCR and does not have enough coverage. Can you have shown the coverages? Yes, you can able to check the coverage, those coverages using Galaxy. All of that you can able to see. Yeah, cut adapt chemomatic when there are ends of the reads have poor quality after doing. Yeah, when we are doing that, we need to do trimming process, right? You can do trimming adapters, how the quality. So you will fix a specific length. That's what we are going to see using our Trimomatic or they have the specific minimum length value, which is 5 to 20. You can able to trim those specific length which is having a low quality rate. It's absolutely that. Yeah, you guys are right. Going into our galaxy, I took so much of your time. Sorry. So if you go into galaxy, you can able to see FastP. There is a tool, FastP all-in-one pre. Just click this tool. Nothing you have need. What you are going to do, either you are going single line or Pardian data. We are going to use a Pardian data. No, Pardian, yeah, Pardian data. So we need to merge. No, it's not merge data. We need to give adapter options, all those things. And we need to give the adapter, whether they have adapter sequence, known sequence, you need to give the adapter sequence input 1, input 2, and all the information what we have need to mention. You need to give those and then you need to run it. Similarly, another tool what I have told is called Trimomatic. It's called flexible for retrimming for Illumina NGS data. So if you mention it, you need to mention you're using a single or Pardian data, or preferably you're using a Pardian data, separate data. We need to give a read one data and read two data. This is a FASTQ read file, and just we need to run it. The information. You can enable the trigonometric operations where you will have the sliding window information, the number of bases of average across the total quality range. So the quality, if it to 20, it means that to it you can see the minimum length. The minimum length is 20. It means the quality below 20 or get into a cutout, that is called minimum length of the reads to be kept. Then of the sample which is less than 3, which is need to be cutted or trimmed. So it's a trialing end, from the trialing end, from the 3-end, that the things are need to be get downloaded. So this is the standard parameter value. We can use this and we can run it. The qualities will get cutted and it will provide the cleaned quality to perform the analysis to do a further thing. Is that fine? You got it? The Trimomatic is only for Illumina data. On the next, FastP, Cataract, all for all kind of data. In the MULTIQC also, you can able to perform those analysis. We can't use it with the 1 GB because when the input data is 1 GB, the output will be provide more information, more amount of larger data that will you see from the next sessions. So the samples, it takes more time to load also, more upload also. So that is not possible. The larger size of genome data were more feasibility to perform with the Galaxy, sorry, Linux only, Linux system only. Okay, let me summarize my slide. So far we have seen in today's session is about raw sequencing data, FastQC where you identify the quality issues. MULTIQC will summarize the quality across multiple samples. Another tool which is called FastP, Catadapt, Thermomatic, where it just helps to remove read quality data. And then host contamination should be removed before consideration. At the end, we'll have a high quality pre-processed data to perform the downstream analysis like genome assembly, variant reduction, taxonomic identification, annotation, everything that we possibly into a much bigger, wider way as an output. And this is the heart of an analysis. Once this has been done, the next steps has been taken forward, or else we will be stuck in this step and you can't able to perform the rest later. And here I have your references. What are there is a book which is edition from Springer. There's this next generation sequencing and data analysis, which was help for a beginner to learn. And also we can able to see bioinformatics data skills, genomic analysis, fast QC user documentation, multi-QC documentation, another tool documentation to understand it into a much bigger and wider way to how things are things. So this is all about today. I have take almost 2 hours of today's sessions. I hope you guys have learned so many things from my lecture.